present here a manuscript that I think will hopefully be one of many that we receive at the journal, and that is a clinical trial design paper. There's a few homes that have been historically placed for these to be published, and we think that this paper hopefully will be something that will set a trend. So I'm privileged on behalf of my co-investigators of the LIFE-BTK study to present the trial design paper for the evaluation of a drug-eluting resorbable scaffold in infrapopliteal arteries. This is the design methodology for the LIFE-BTK clinical trial. I am a co-principal investigator of this study that is sponsored by Abbott Vascular. I think everyone's aware that 1 to 3 million Americans suffer from critical limb ischemia or chronic limb-threatening ischemia, with amputation rates that approach 40% at 6 months, with half of many amputations being done without any vascular evaluation. These patients are highly comorbid and have a high mortality of up to 25% in the first year, over 50% at 5 years. And by having a diagnostic angiogram, the risk of amputation is reduced by up to 90%. Treatment for the below-knee circulation has been complex and has been confounded by a number of biological and clinical challenges. Bare metal stents, angioplasty, drug-eluting stents, drug-coated balloons, and atherectomy devices have all been tried, but we still have the unmet clinical need of needing inhibition of intimal hyperplasia, scaffolding to resist recoil, and eventually the ability to leave nothing behind to preserve surgical options. So, to wit, the option of using a bioresorbable drug-eluting scaffold that's a temporary scaffold that will fully resorb, but that can also provide effective anti-proliferative therapy was something of a pipe dream at one time and is now had been actualized. You may recognize this as a evolution of the prior absorbed BVS. It's a PLLA bioresorbable scaffold background with a coating that's comprised of everolimus and a bioresorbable polylactide PDLLA with 4 platinum markers on either end for visualization on a monorail coronary-like drug-eluting stent delivery system. The LIFE-BTK trial, which I'm privileged to be a co-principal investigator with Ramon Varco from Australia and Brian DiRobertis also from New York. We have enrolled 261 patients globally in a 2-to-1 randomization of the SCAI-BTK bioresorbable scaffold versus PTA, with the objective of evaluating safety and efficacy compared to angioplasty in the planned treatment of the infrapopliteal circulation in patients with critical limb ischemia. The primary efficacy endpoint is going to be measured at 12 months with patency plus limb salvage as the endpoint, maybe something as a point of discussion. And the primary safety endpoint is major adverse limb events plus perioperative death at 6 months. Overall, we'll have 5-year total follow-up. This is the diagram of enrolled patients. About 20% were enrolled in the Asia Pacific region and the remainder in the United States. Again, we've highlighted, and I think for the purposes of the design, there have been innumerable designs for clinical trials in the below-knee segment, all of which have been confounded by challenges with adjudication of the endpoint. We've settled on limb salvage plus primary patency, which is freedom from above ankle amputation in the index limb, freedom from total occlusion of the target vessel, or binary restenosis as measured by Doppler ultrasonography, as well as freedom from clinically driven TLR at 12 months. The null hypothesis is superiority with a one-sided alpha of 0.025 against balloon angioplasty. The assumptions that we made were primary patency of PTA would be approximately 55% and the SCAI-BTK 75% with a treatment effect of approximately 20%. With respect to the safety endpoint, this is somewhat less controversial and is a composite of major adverse limb events and perioperative death. Limb events equates to just like MACE. It's a similar assessment of above ankle amputation in the index limb or major reintervention at 6 months. And of course, perioperative death is mortality within 30 days. And here, a one-sided alpha for non-inferiority, also of 0.025, with assumed rates of approximately equivalent 95% efficacy. Importantly, this trial for a peripheral trial has a relatively aggressive powered secondary endpoints. These include binary restenosis of the target lesion, again using Doppler, with the assumed rates as noted below with a one-sided alpha of 0.025, and freedom from above ankle amputation, total occlusion, and CDTLR also with a one-sided alpha of 0.025, and the assumed rates are noted below. All patients that were included will be Rutherford-Becker 4 or 5. Rutherford 4 is rest pain, 5 is minor tissue loss. They'll be treated in the proximal two-thirds of the native infrapopliteal arteries, avoiding the ankle zone. Maximum of 2 de novo or restenotic from angioplasty lesions are included with greater than 70% diameter stenosis. The reference vessel diameter will be 2.5 to 4, consistent with the size matrix of the drug-eluting scaffold, and the total scaffold length will be a respectable 17 centimeters in toto at maximum. This is the sort of schedule of events. The baseline assessments include the usual demographic and medication history assessment with laboratory assessments. Importantly, patient-reported outcomes will be an important part of this trial, and I think will be paradigmatic in this field going forward. Hemodynamic assessments are compulsory, and wound assessment is being done with a camera-based system and with a wound care core laboratory. Also, I think what will become the de facto standard for future trials in the space. Other angiographic assessments will, of course, be made, and endovascular imaging is encouraged, if not required. For the procedure, patients will be on antiplatelet therapy. They're randomized 2-to-1 after evaluation at angiography, and then PTA or scaffold is done in a blinded fashion to the patient. Treatment will not permit the use of atherectomy or scoring or cutting balloons or lithotripsy, and pre-dilatation at a ratio of 1-to-1 is required with a residual of less than 30% to be randomized. Post-dilatation is required, and successful treatment is defined as final diameter stenosis less than 30%, final number of runoff vessels at least as well as seen at pre-procedure with no residual dissection or embolization, and no other complications on angiography. Follow-up is done in a very pre-specified and aggressive timeline consistent with the need for these patients with critical ischemia, 30, 90, 180, and 365 days, followed by annual follow-up thereafter. The wound will be assessed also fastidiously by photography and the wound core laboratory, and Doppler ultrasonography with a very detailed protocol to assess the primary endpoint will also be completed. In addition, there is a pharmacokinetic substudy that will enroll a total of 7 subjects and will also be included as part of the FDA requirements for the IDE. And again, blood collection will be completed in these patients at time 0, 10, and 30 minutes, and hourly thereafter, up to 2 hours, and then 4, 6, 12, 24, and 48 hours out to 60 days. One of the most important things I think about this space is that patients with chronic limb-threatening ischemia in the United States tend to be underserved, and we have gone above and beyond, I think, to recruit both diverse patients as well as diverse investigators. Every aspect of the trial is core lab adjudicated, from angio and intravascular imaging through duplex and to wound care, with what we would argue are leaders in that appropriate respective field. Vessel preparation, sizing, and post-dilatation are all algorithmically pre-specified. And again, I think what's particularly important is the assessment of the wounds by our core lab. So we hope that this study will serve as a paradigm for moving forward in this field. As yet, there are no large-scale randomized controlled trials that have demonstrated successful effectiveness and safety of a drug-eluting device in the baloney circulation, and we're hoping that this will be a harbinger of things to come. I thank you for your kind attention.
All right. Well, congratulations on just an amazing study design and really comprehensive endpoints, and the PK study is going to be of interest, as you said, because this technology seems to hold, promise to avoid the potential complications from DEB, which is downstream microvascular obstruction. But I was just wondering about training that the operators are getting in terms of how to use these scaffolds and preparation of the vessel, what's required?
Yeah, so as you can imagine, given our coronary experience with bioresorbable scaffolds previously, it was obviously clear that aggressive predilatation, which is really specified here as standard of care balloon angioplasty, but specifically with optional imaging, although I think many operators chose to use imaging, and also one-to-one sizing to the best of our ability. We, in fact, sent a pre-test to all investigators to see what their ability was to size vessels, and we aggressively trained them on appropriate sizing because we tend to habitually undersize below-knee intervention. So we've done our best to train what is a relatively experienced set of investigators on appropriate preparation, but it's balloon angioplasty to keep the field level. Once you introduce additional vascular injury, the risk of intimal hyperplasia is greater, and we thought that it would potentially negatively impact the balloon-only arm if no drug was being applied. In addition to that, post-dilatation of scaffolded patients is compulsory, likewise, ideally with an image-optimized sizing algorithm. We did not ask, we specifically precluded bifurcations, and we made sure that adjacent areas that were injured were all covered with scaffold if they were randomized to that group. So we've done our best. I can also say that we as PIs have reviewed every angiogram and have personally counseled any concerns that there may be with operators. So, we've invested incredible time and attention from ourselves, the core laboratories, and every other possible mechanism. A couple of other unique characteristics were with this difficult population and with the pandemic, which, it's not great to launch a complex global trial in the middle of a pandemic, but kudos to the sponsors for helping execute it. We had a SWAT team of extramural support sonographers and research coordinators that were able to make home visits to the patients if they were unable to come in for follow-up. And so we're doing a great job with follow-up duplex, particularly because duplex is the way we're adjudicating the primary endpoint.
And Dr. Sahil, being a peripheral expert on the panel, what are your thoughts on the trial and how it's going to impact, how the results are going to impact care?
Yeah, I think this is the trial of the year for the peripheral space. I'm involved in the clinical events committee in this trial, so I have a small role, but big kudos to Sahil and Brian and Ramona as well as Abbott for taking this on. And maybe I'm actually going to ask a question to Sahil, and just for our space. So if you're not familiar, if you're not in this space very regularly, we've had 2 below-the-knee failed U.S. trials for drug-coated balloons. We've had a failed stent trial last year. If this fails, what would you have done differently, and if anything, and what do we do differently moving forward, or are we done? And I know it's a hard one to ask going into the
Yeah, well, we don't know the data yet. We're hoping that they'll be presented this fall. We'll be approaching that data lock sometime at the end of the summer. So I think it's hard to imagine with a failed trial what would happen to the space, but it will certainly be a setback. So we're hoping we don't have to think about that eventuality. But I think certainly the challenge is predominantly in the trial design, which is why I think it's great that this design is being published in JSCAI and hopefully will be widely reviewed and examined and critiqued, I'm sure. But one of the unique aspects is looking at binary restenosis by Doppler in the below-knee circulation. No trial has had the courage to do that before. We're fairly confident from our preliminary discussions with core laboratory experts and other experts in the field that it's tenable. There are predicate smaller studies that have done that. And it allows us to have the fidelity with a smaller study. The coronary trials are 10x what we do in peripheral, largely because of complexity and the need for long-term follow-up. And so with a 261-person trial, we're hoping that we'll find the fidelity using this new endpoint. We've had detailed discussions with FDA, and I think FDA is very sanguine about the fact that we've had no wins in terms of clinical trials in this space, and they recognize that this is an incredible unmet need for this population. So we're hoping that this design really is a way forward. We also think the wound history that we're going to get from this is completely unprecedented and will tell us a lot about the natural history of CLTI.